Okay, looks like we are starting. Great. All right. Yeah, so welcome everyone. This is the ninth ASIP SciTech Impulse webinar. The 8 previous ones were all on the topic of modeling and simulation, but with this webinar, we decided to broaden the topic to the more general topic of industrial biotechnology, which is the competence of ASIP as a center, as a whole center, right? So today we have a really interesting topic. It's on microalgae and how we can use them as sustainable cell factories. We have two very exciting speakers, Rita Mota, who is actually from ASIP, and Muriel Bador from University of Rouen. So as you can see, the meeting is being recorded, and there will be 2 presentations. First, Rita, about 20 minutes with about 10 minutes for Q&A. And then in the Q&A, I hope we have the settings right this time. You are either able to ask questions in the chat or either anonymously or with your name. And you can also raise your hand and we can call you into the podium if you want to ask a question live. And then after an hour, we will stop the recording. So I think that covers the housekeeping. With that, I will already introduce our first speaker. Dr. Rita Mota. She's co-founder and CEO of Cyanocare. She's also a senior researcher at ASIP at BOKU, where she has been for several years. And I think you started Cyanocare last year. So I'm actually really excited to see what Cyanocare is up to. With that, please go ahead.
Yeah, thank you very much. Good afternoon for everyone. So let's try to share. So are you doing my presentation? Oh.
Yeah. Okay. We can see the screen. Yeah, perfect.
Yeah, perfect. So exactly. So we're going to talk a bit about microalgae. And in my case, I will give an example of Sanofarm. That's what I was working with. So I will present you that. But first of all, just to make sure that we are all more or less on the same page. So when we talk about algae, so algae is just really like a common name, but it includes a lot of different organisms. This is the ninth presentation for the European Algae Biomass Association. So we can be talking about cyanobacteria, microalgae, seaweeds, but yeah, basically they can all perform optogenic photosynthesis. So they can use the visible light to fix CO2 with the release of oxygen. We can also usually have chlorophylls as main photosynthetic pigments, and they lack of differentiated tissues as the plants have. And they can exist everywhere, but they are particularly important in the aquatic ecosystems. And basically, they have a huge potential, and this is nowadays really being discussed, especially in Europe. That's where we are. And well, mostly you are eating all the time sushi. Maybe you also try spirulina, but that's not only for food. So there's really a lot of other sectors that also are already being commercialized. And well, now I'll give you an example of one of the products that can be produced by algae, which is Sanofarm. So we are talking about a specific strain of cyanobacteria. So it has this weird name, Prophosphaerus wackensis CCY0110. But basically, it's a marine unicellular cyanobacterium. So this was isolated for the coastal waters in Zanzibar. And it's very interesting because it produced a huge amount of released polysaccharides, so the specific type of EPSs, of extracellular polymeric substances. So here is a normal optical microscopy picture, but it's actually stained with a specific dye for carbohydrates. So that's what you can see here. And well, I have been studying for many years. I did a PhD on this topic. So basically, I'm trying to characterize it. So it has composed by 9 different monosaccharides, including uronic acid, which is not very common in other types of microorganisms. But also has some proteins and sulfate groups in its composition. They have a very big molecular mass fractions So this all leads us to a very complex and ramified chemical structure that has been very difficult to actually find out the structure. But it might also be that it is a very versatile biomaterial. And so throughout many years, so this is a very long story, but have been specifically focused on biomedical applications, because when I was in Portugal, my previous specific group was basically in health. So we studied different applications, as for example, using Sanoflan as a drug delivery system, and when we can fine-tune the delivery with using the biolactive ions or capsulating. So we test different approaches, depends on the application that you want. We also use Sanoflan as a coating for medical devices. And this, so it's not antibacterial, so it's not killing anything, but it's actually preventing the addition of bacteria, fungi, proteins. So our plan or still is to actually use it for ordinary catheters, because it has a lot of prone to biofilm formation. So we want to avoid the initial step of this biofilm formation. We also eventually move on also for wound healing, and this we are still also working with these in my previous institutes. Yeah, basically it promotes the wound healing process. And we also saw some antioxidant and anti-inflammatory properties. And well, something that was very straightforward to see in all of these applications. Just like when you do solution with Sanoflan, it gets really viscous. So we also studied this behavior, and also the emulsifying properties using different compounds. And then eventually even studied using human blood plasma. So how was the behavior of Sanoflan? Again, always envisions in biomedical applications. And this leads to 3 granted patents already in the EU and 1 in the States. Well, this is also very interesting, and it's already like a long story. But at the end of my PhD, I really started believing that, well, one day Sanoflan could enter the market. Like, why not another natural polymer? And this roadmap, so the biomedical, is for sure not the easiest one to enter. So at a certain point, I shifted a bit to actually being able to use Sanoflan as a cosmetic ingredient. So there is a need, there is a very big market, and then we can have this multifunctionality of Sanoflan, so as well as humidifier and also bioactive agents. And yeah, so well, it's also very important to actually be able to validate a lot of things that have been studied by experts in the field, in this case, a Portuguese contract research organization. So, namely, the safety to use Sanoflan. And this is one of the many tests we can do. So, the ocular irritation potential test has this name because before it was seen in the eyes of the rabbits, but you can also very easily do with X, with the choroidal lymphoid membrane of X. And well, as expected, using different concentrations of Sanoflan, there was no effects whatsoever. And so eventually we could move on to in vivo studies. So it's called a patch test. Humans would like for 48 hours in contact with these ingredients, and then you can also see if any symptom occurs. And well, as expected, again, nothing happens. So we can actually state that Sanoflan is biocompatible, and we can proceed with studies. Something that was also validated by this very nice in vitro platform. So it was like 30 different assays with skin-related cells. So this is just one example. This was done recently. So this is one of the examples involving the skin aging. And what we could see is like actually the use of Sanoflan, again, in a dose-dependent mechanism, which is normal for this type of polymers. There was actually the inhibition of transcriptional activity of this activated protein. And yeah, so this is very interesting because so this one of them involved in the skin aging assays, like both those stuff is very hormones, so there's a lot of effects here, and so we can use this molecule to actually inhibit production. So basically, with all these several studies, all these together, we could actually validate the anti-inflammatory activity of Sanoflan. And it seemed to be by a photoprotective mechanism, but we need further in vivo studies to actually conclude this. But this was already very interesting. And so eventually we could move on. So because until now I was also only talking with Sanoflan in solution, so Sanoflan alone. And so of course, eventually we also move on to mix Sanoflan with other common cosmetic ingredients. And then we had to do a lot of studies that are used in this field. So you see how the viscosity changes, it's stable throughout the time, different temperatures. I mean, in terms of color, aspects, odor, also pH. So all of these are normal studies that needs to be done for the cosmetic ingredients. And always we saw this typical behavior. So this is, so we have increase in viscosity with the higher concentrations or if we mix with other ingredients, and then we have this decrease when we apply the shear stress throughout the time. So this is very typical for this type of polymers and is called non-nucleated cellular behavior. In some cases, we actually saw our alteration. For example, in this case, we developed cations as we were already expecting, and also if the pH changes, we can also saw an increase in viscosity. And this is mostly due to ionic cross-linking or hydrogen bonding. So again, very typical for these polymers. And we can fine-tune this depending on the final properties that we would like to see in the formulation that we are developing. And we can also see some synergistic effect with other thickeners. So we add Sanoflan together with synthetic ones, for example, carbomers, or natural ones, so xanthan-gum, for example, we see a very high increase in the viscosity. So again, we can all fine-tune this depending on the final properties that we want. And so we went to actually develop our own formulations as a proof of concept. It's still ongoing, but this is basically we try to develop always two different formulations to compare. One more synthetic based ingredients, another more natural based ingredients, always with Sanoflan. And then again, this is all normal tests that we need to do. So thermal stress, when we change the temperature, we also freeze thaw, different pH, centrifugation, what happens to the final formulation. And well, as I said, it's a work in progress, so we still need to optimize this, but in general, it was actually very stable in terms of aspects, other pH, but the viscosity changed a bit, so it's not really 100% stable. So we are actually fine-tuning the concentrations of the different compounds, also Sanoflan. And as you can imagine, there's a lot of different possibilities, so we still need to work. We are working on this at the moment. But we had the opportunity to actually perform a sensorial evaluation, so this was very interesting. And actually, the results were very nice. So we compared these two different formulations of Sanoflan with a premium commercial formulation available in the market. And we had a very positive feedback using Sanoflan, especially in a natural-based formulation, and in particular regarding the hydration feeling in the skin. So there's definitely something that we want to pursue. But of course, we have always to constantly produce Sanoflan, obviously. And so we are also trying to optimize this production. And this was in collaboration with Naltazar and Inventors. So these are like two companies that had listened already before, if you attend the previous webinars. So I'm not really explaining that part, but so we together we design with the design of experiments when we change some of the settings that we can change regarding our lab scale production, namely the light intensity, because it's very important. It's the most important factor for this strain. So we vary the light intensity using LEDs, but also the steering speed and also the temperature, while the aeration and the culture medium was stable, we didn't change. And then we did, so it's the lab setting that we have, and then we do like measurements throughout the time. So growth, so the optical density in a chlorophyll A, but also we quantify the carbohydrates. It's what we want at the end. So we want to understand the production of SANOFLAN. So we quantify for the whole culture or for the released polysaccharides isolate. And yeah, so with the help of these companies, we also perform computational fluid dynamics. So we try to really understand what is the impact of the light intensity on the cells. So this is almost finishing, but we still don't have everything. So we are starting to write a publication on this. And so we had also like onsets inside of the cultures that we could monitor how was the penetration of the lights. And this is always to also help the scaling up of the process. This is lab scale, and we want to go into industrial scale one day. So all this information, the final goal is to develop this digital twin. So this is just an example. It's not the final one, but here we can change so the range of temperature, vegetation, and the lights, as I said. And we can see here so the carbohydrates and the optical density and how it varies within this range. So it has been very interesting, very different, very interesting results, and we are just finalizing this. And then, of course, this is just the upstream, and then we have to isolate the SANOFLAN. So just by simple centrifugation, because we need to separate from the cells, because SANOFLAN is a supernatant, right? And then we concentrate the supernatant and we have to remove the salt from the media, and then a dry step. It's a very normal and common downstream process. And then also some years ago, we also, so I mean, I'm always trying to talk about SANOFLAN, but the cells can be also very interesting, right? So we also check the nutritional potential of the biomass, which has a lot of similarities with spirulina biomass, which you probably know about it. So a very high percentage of proteins, also some lipids and carbohydrates. And in this case, in particular, of this strain, was a very high amount of the vitamin A and E, and some of the minerals. So this can also be very interesting. And also that you might also know from spirulina, this strain is also, the cells are also producing these blue pigments, the phycocyanin. So maybe you have already tried blue ice cream or smoothies. So yeah, this strain also produces this phycocyanin. So yeah, this was basically it's a story that is still in progress, but I hope you can envisage the potential of SANOFLAN as a new cosmetic ingredient, because yeah, we do. So that's why we created SANOCare. So just last year we got the ABS pre-seed funding, and so we are just we just officially just did this since December. So this is very recent. And what we are doing at the moment, and these already arrived last week here in Toulon. So we are starting the pilot scale production. So we start this 100 liters for the bioreactor, and this will allow us to constantly produce a lot of SANOFLAN to finalize all these tests, and also to start approaching companies. Well, actually, we were already approached by companies, and we're going to be able to deliver more compounds. And yeah, of course, there's a lot of people have been involved in this project before and also now. And yeah, thank you very much.
Yeah, thank you, Rita. That was a really cool talk. Very, very nice technology you have there. So are there any questions from the audience? Yeah, so please raise your hand or ask a question in the chat. Apparently, I need to open the Q&A panel, otherwise I don't see it. Okay, there's no questions there yet. So this photoreactor that you showed, yeah. So how does it work? So there's actually is it like a plug flow reactor where the cells are continuously pumped around, right?
Exactly. So it's a tubular one, and then yeah, they are continuously pumped around. So it's a closed one. It's very important for these type of strains to avoid contamination, because you know a lot of sugar outside the cell, so it is very prone to contamination. So we need to have a closed system, and then also always with aeration to mix and to make sure that all the cells have contact with the light and the nutrients, and this of course we're going to have to fine tune the best conditions for this reactor in the room that we have access.
Okay, we have a question from our other panelist. Yeah, so Muriel,
Thanks,
please
Peter,
go
for
ahead.
a very interesting talk. I was having a question because you mentioned that your polysaccharide was bearing a lot of sulfate group. How do you manage to control the reproducibility of the sulfatation from one batch to another batch, which is normally quite tricky?
Yeah, well, that's true, but actually throughout all these years, I never had any alteration actually. So this is quite interesting, this strain, luckily. It's not really, I mean, luckily, so even if I wanted to change it, it's very difficult. So so far I don't see alterations really, and I had producing in different places, and characteristics is the same.
Thank you.
So it's completely natural.
We have a question by Barbara Petschacher. Let me bring you on screen. You should be able to ask a question live. Yeah.
Hi, yeah, I managed.
Hello.
Thank you, Rita. Very interesting. I just wanted to know, are there any regulatory issues you already have to take account of at this stage of your research?
Well, yes. So the safety ones were the most important ones, just to make sure that there's no toxins, for example, because there's something that, well, in the cyanobacteria cases, it's actually not that common, but unfortunately, you know, there's bacteria on the name, so people get think that we also have toxins, and so so this biocompatibility was very important to make sure. That's basically what we have to provide. But then we still didn't register as a new ingredient, actually, something that we have to do. But we wanted to establish the industrial production first, because it's very important to establish that, and then we can register as a new ingredient. And that hopefully that will gonna happen during this year.
Okay, thank you.
Okay. Okay, actually, following up on that. So initially you tried to use this product for wound care, right? So that's medical devices regulation, right? And now, can you maybe go a little into the differences in market entry that you observed between cosmetic products and medical devices?
Yeah, well, definitely. So from a regulatory point of view, it's completely different. So of course, we might go still in that direction if everything goes okay. But yeah, even just regarding the production, we need to follow all the regulations of the production. But at the moment, we cannot. So we don't have access to actually like manufacturing productions that complies with a medical device. So that's very tricky. So it might be possible if we see the needs into that direction, but yeah, need to be a way controller environment in terms of production.
But so it's still there's still a lot to do for cosmetics, but then for medical devices it would be it was okay.
Yes.
But then once you have this established for cosmetics, it might actually be much easier, right? To
Exactly.
go there
That's
for medical
the
devices.
plan.
Okay.
Exactly. That's
Okay, that makes sense. Yeah. And so you observed this change in viscosity over time. Did you figure out where it's from? Is it degrading or?
Yeah, not yet actually. So that's how just I have like two master students starting just now on this topic.
Okay.
Because so I'm a biologist, so about algae I know. Okay? About formulation, chromatic formulation is not really my my field. And when I start checking on it, there's like so many possibilities, you know. Of course, we are thinking of like skin aging, like regarding with anti-aging creams, because it's what we already know that cyanophlan can have an effect. So also with hydration, so it's not that it's already a bit fine-tuned, but still there's a lot of different ingredients we can use and different concentrations. So this is, I mean, if someone is present that is expert on that and want to help me, we can collaborate together. That for me has been quite difficult to actually decide. And every time we want to try something, you know, we use a lot of cyanophlan because and a lot of time because we need to do all these cycles of temperature. So it takes it's a very long process And then at the end, if I find out that it's not really stable, so I have to start from the beginning. So it's work in progress.
Okay, yeah, but I saw you had a lot of interesting collaboration partners in the last 2 years, but that's quite nice. Yeah, so I don't know if there's any, doesn't look like there's any more pressing questions. So actually, I was happy to see that that the topic of modeling and simulation cannot that we cannot escape it, right? So yes, yeah. Novasign have been on this webinar before, but Simvantage actually will be on the webinar in April. Is the current plan. So let's see. But I think it might be a different topic. But so they did computational fluid dynamic simulations then of this tubular reactor, or
no, it was from the lab scale. So it was from
the lab scale.
Yeah.
Okay. Okay, and then, but
Yeah,
then for scaling, I mean, that collaboration could continue, right?
Yeah, exactly. So that's very tricky, right? Because also the geometry varies a lot, and that's another challenge that we have.
Yeah.
But it's just not possible to do it another way. And so it's still valid, all the information that you get from the lab scale.
Yeah, of course. I mean, now once that is well characterized, it's much easier to
Yes, exactly. But we have
scale
to
up to
start
something else.
somewhere, and the lab scale,
Sure.
yeah, we have this class. This is the easiest thing to have. I was already used to work with it, and we are always limited also with the conditions that we have in the lab. So yeah, I would like to have we can, and there's so many options. Also just regarding the lights, there's so many LEDs and setting of lights that you can have, but
Yeah.
you have to start somewhere with the conditions that you have, and it's completely still valid. So we are
It's quite unique, right? It's an interesting challenge.
Yes, also for them was also the first time to work with phototropic organisms. So it's not that common. It's not that many people working here with algae. So this was another challenge also to understand and to think how we can use that data, right? And then they have all this modeling behind that, no idea, and all these formulations that we need to understand how this correlates, right? How we can correlate the growth with the cell and the measurements that we do and what is happening inside. So this has been also quite challenging for us to how we use proper this data and how we can extrapolate for the scale-up process. But
yeah.
I hope this year we're already gonna publish this information. So then,
very nice. Yeah, really exciting. So if there's no more questions, yeah, if you have any more questions for Rita, please write them in the chat. We can also answer them later. But with that, yeah, thank you for coming. And with that, I will introduce our next speaker, Muriel Bardot. She's professor at the University of Rouen and the CEO and CSO of Alga Biologics. And Muriel was also the co-supervisor of my PhD thesis. Yeah, so we met each other in Singapore at the Bioprocessing Technology Institute. It's been a few years, yeah, but I'm very happy to have her on here. So with that, please. Unfortunately, yeah, the camera is not working, so I cannot see you.
But it's still not working?
No, unfortunately not. Yeah, but then yeah, I will.
Sorry about that.
I will share your slides. Yeah, so you should be able to see them now. And with that, I hand over to you, Muriel.
So thank you very much. I want to thank you very much, Nico, for the kind invitation to present some of the work that I have done with my team on the last years. So the story that I will explain to you today are starting actually a few years back as well, when I moved back from Singapore to France on my professorship position, and where I starting a completely new project in the lab. So if you go to the next slide, so the story has started in the lab at the University of Rouen-Normandie, which is located, as you can see, in the north of France, in Normandy, at the University of Rouen. And the lab is specialized in plant glycobiology. So maybe you can put everything on the screen, Nico, it will be easier. Thank you. And in this lab, so we are interested in the structure, the biosynthesis, and the functional property of plants and macroalgae glycoconjugates. So like Rita, I will talk a little bit about sugar, but for me, I will talk only about sugar which are present on proteins that we call glycoproteins. And some of my colleagues from the lab, they are also working on the polysaccharides, and but more on polysaccharides from plants, and they are investigated their structure function relationship in relation to the reproduction of the plants, as well as the plant immune system, and especially focalizing their research on the roots. But myself, I was always interested in the synthesis of glycoprotein. And when I moved back to Singapore, I started to look at the synthesis of glycoprotein in macroalgae, as at that time, almost nothing was known on these topics. And I was interested to see whether the protein and glycosylation, which is occurring in the endoplasmic reticulum and in the Golgi apparatus, were similar to what we know about protein and glycosylation in plant, or whether it was different. Because previously I had been working on the plant and glycosylation. So when I started the project, we started to look at which macroalgae to choose. So for us, we are interested in the protein glycosylation of macroalgae, which are unicellular. And in fact, when you look at the repartition of a macroalgae within the tree of life, you can see that they are present in very different phylum. There is a huge diversity of macroalgae, and they have a huge capacity of adaptation, which means a very diverse metabolism. So I must admit that when I started this project, in fact, as we were not specialized in macroalgae, we didn't know which one to choose. So we decided to take several macroalgae representative of different phylum, and we start extracting the protein from this macroalgae, analyzing their N-glycosylation, and see which kind of structure we can find on this macroalgae. And in fact, we were very surprised about this result, as we found a very huge diversity of N-glycosylation on the protein of the macroalgae, which is summarized here. So of course, I will not enter into the detail, but if you focus on the green square within this figure, you can see that if we look at some of the macroalgae like the chlorophyllaceae, which belong which group belong Chlamydomonas, which is a quite well-known macroalgae. In fact, we found some glycan that has never been discovered before. We found some linear glycan, which were bearing some methylation, which were bearing a lot of xylosylation motif. And it was very, very bizarre to find this kind of structure. And if you look at all the other macroalgae that has been studied so far, they are all bearing different structure of glycan. So this is still an ongoing field of research that we continue to work on in the context of a European collaborative project, which is called GlycoN, which is ongoing. And we have currently a PhD student, which is working on understanding the role of the different enzymes which are involved in the glycosylation pathway of macroalgae. When we did this screening, actually, we were quite lucky to find one particular macroalgae, which is called Phaeodactylum, which is a diatom and which is living within the seawater. It's a marine macroalgae. And if you look at this picture, in fact, what we found in this macroalgae was protein which were bearing oligomonocytes, with exactly the same kind of structure that what you can find on some of the protein from mammals and human cells. So of course, as we were interested to try to produce some recombinant protein within macroalgae, we decided to focus especially on this particular macroalgae in order to express some biopharmaceutical within this macroalgae. So based on the work that we have done previously with Nico in Singapore, which was focusing on the production of antibodies in mammalian cells, of course, I decided to start by producing some antibodies within Phaeodactylum. So if you go to the next slide. Thank you, Nico. So the first antibody that we produced in Phaeodactylum was in collaboration. Again, it was in a European project, which was in collaboration with the University of Marburg in Germany, as well as with the St. George Hospital in London with Professor Julian Ma. And on this work, we produced an antibody, which was the sequence of a human antibody, which was able to recognize hepatitis B virus antigen. So we demonstrated that Phaeodactylum can produce successfully these antibodies, that the antibodies was correctly assembled with a wide shape. And of course, we proved that this antibody was glycosylated on the Fc part of the antibodies. And as you can see on the slide, we found the same type of N-glycans, which mean oligomonocytes that we had found on the natural endogenous glycoprotein of the macroalgae. We, of course, demonstrated the functionality of the antibodies. At that time, we demonstrated in vitro. We demonstrated that the antibody was able to recognize the virus and neutralize the virus. And we also demonstrated in vitro that it was able to bind to different gamma receptors from the human immune systems. After this, so this was the first demonstration that we can use macroalgae to produce antibodies. Later on, our colleague from the University of Marburg produced another antibodies, which was a chimeric antibodies targeting another viruses, which is called the Marburg viruses, which is like a kind of Ebola virus. It's similar to the Ebola virus. So they demonstrated that they can produce some chimeric antibodies which were bearing an Fc part from either rabbit or human, and both antibodies were successfully produced in the macroalgae. And again, they demonstrated that this antibody produced in macroalgae was able to recognize the virus, the Marburg virus. So based on this result and on the different patents that we had on this technology, we decided with the University of Normandy and some of my colleagues, Dr. Malika Mekalfi and Professor Patrice Lerouge, we decided to create 4 years ago now, Alga Biologics. And of course, as you can guess, the objective of Alga Biologics, if you can go to the next slide, Nico, is to harness the natural power of marine macroalgae as a cell biofactory to produce sustainable, safer, and more affordable recombinant protein. And we are now focusing on the production of 3 types of protein. Of course, we are still working on antibodies, and that's the result that I will show to you later on. But we are also producing right now some cytokines, which are more and more used for research as well as for cosmetic application and as biopharmaceuticals. And we are also producing some peptide for the cosmetic and pharmaceutical application. And what we want to develop is a new way to produce such kind of recombinant protein, because currently they are mainly produced in mammalian cells or in bacteria for some of them, which is quite expensive. At least with mammalian cell, is very expensive, quite complex, as the culture media that we are using are complex. It's consuming a lot of energy, and it's polluting, as there is a lot of emission of CO2. And we want to secure also such kind of production and avoid any risk of viral or endotoxin contamination. And in fact, the macroalgae are solving all these problems while they are more sustainable for with a lower environmental footprint. And we can also make this recombinant protein in a GMP compliant process. So we want to be part of a future and propose such a sustainable bioproduction. So far we have been producing different type of protein, but today I want to focus on 2 examples. One example is the trastuzumab that we have produced in macroalgae as a research and diagnostic tools, and I will show you the result that we obtain. And I also want to focus on one another antibodies that we call neuroblastoma, which is our demonstrator to reach the pharmaceutical market, and which is targeting a pediatric cancer called neuroblastoma that affect 25,000 kids worldwide, and which is today treated with one antibodies which is produced in mammalian cells, which is not very efficient, as 50% of the kids are still dying from these cancers. So if we come to the result of the trastuzumab, so we took the sequence of the trastuzumab, the exact same sequence that the one which is produced and commercialized from mammalian cells. We produce it in the marine macroalgae Phaeodactylum, and then we tested the efficacy for different research and diagnostic application. So as you can see in this figure, so we compare the efficacy of the trastuzumab produced from macroalgae with the one produced in shore using FACS analysis. And as you can see, they present the exact same behavior and capacity to recognize the HER2 antigen, which is the target of these particular antibodies. We prove that it was working efficiently in Western blot, and the specificity of the antibody was good. With our colleague from the hospital in Rouen, we demonstrated that we can use in order to do some immunohistochemistry on breast tumor tissue that you have here, and we have a specific labeling of the tissue. And these antibodies are also being tested by one of our partners, which is Polydetect, which is developing some ELISA kits. And as you can see, it was also successful in their ELISA. So through this application, we demonstrated that the antibodies that we are producing in macroalgae can be used for research and diagnostics. But of course, what we want to reach in a few years from now is the biopharmaceutical markets. So we produce these antibodies, which is targeting the neuroblastoma in macroalgae. And here you have some example of the result that we obtain after producing it at 200-liter scales, which is the scales that we reach already in the company. So we demonstrated that this antibody is able to bind to the antigen, to the neuroblastoma cells. We demonstrated that they are able to bind to the receptor from the human immune systems. And recently we started to do some tests in vivo. And as you can see here, we generated some xenograph of neuroblastoma in zebrafish. So this is the D0 that you get here. You have the tumor which is highlighted. And 3 days after injected the antibodies produced in macroalgae, you see that the tumor is decreasing and you only observe some small spotting. So we are continuing the characterization of these antibodies, and we are currently testing the pharmacokinetics of these antibodies, and we are also working on the understanding of the mechanism of action of these antibodies. So today we are able to do the cell line engineering and produce any recombinant protein of interest. But so we are generating some stable cell line with our macroalgae, and then we develop the process, and we go from lab scale up to 200-liter scale right now. And our next step is the same as Rita's next step. We want to increase the production size, and we want to go up to 2,000 liters. We are also working on the improvement of the production yield, and of course, at least for the pharmaceutical market, we need to implement such a production system in a GMP regulation. So we are currently a team of 8 persons working together with me with different expertise. Some people have expertise in the pharmaceutical industry, others in cosmetics, and others in marine biotechnology. And we are backed also, if you go to the next slide, with different experts which are supporting us either scientific experts or strategic experts, which are helping us in the development of our business strategy. So I would like to thank you very much for your attention. I apologize for not having my camera working. I don't know what is happening, and I'm ready to answer your questions.
Great. Thank you for the nice talk. Now, this was since we had a lot of IT issues today, so I also had to do the slides on the side.
You did good.
So
You did good, Nico.
yeah, thank you. But yeah, I hope there's questions from the audience. So please feel free to ask away or raise your hand. So I was let me see. I wrote down some questions. So yeah, this neuroblastoma antibody. Actually, I also worked on one. Yeah, dinutuximab, together with the company of Pyron. Actually, when I came back from Singapore. So what are the sequence? So are the sequence changes, or is it a completely new antibody, or so?
We have both in hand. In fact, the result that I showed you is exactly the same sequence as the antibodies developed from Apeiron, which is commercialized in Europe under the name Qarziba. So we took exactly the same sequence, and we also have a version where we introduced some mutation in order to try to improve efficacy. So actually, we are working on both at the moment. But I don't know, the version which is having exactly the same sequence, we are discussing right now with the French regulatory agency in order to understand whether we are agreeable to call it biosimilar or not.
Okay, because
as
of
we
the
change
different
the
hosts.
expression system, it's so this is not clear yet.
Okay, and so this mutation, this is in the like in the antigen binding domain, or in the does it concern the effector functions?
No, it's more on the FC part.
On the FC part. Ah, okay, interesting. Yeah, and then now I'm actually curious. So what is the titer that you can achieve with these microalgae at the moment?
So this is for the moment the bottleneck that we still need to improve. So right now we are above 10 milligrams per liter, which is still which is still low, but just to make you understand from where we are starting, when we did 2 years ago the first batch of production at 200 liters, we were only having a few hundreds micrograms.
Okay,
so we did improve quite a lot already in 2 years, going from the microgram scales to more than 10 milligram scale, but we still need to improve. But we know that we do not need to reach grams per liter, for example, which is the current standard with the mammalian cells, because we are so cheap as compared to mammalian cells. But as soon as we will reach 100 milligrams to 200 milligrams, we will be at the exact same cost as what is the cost today of antibodies produced in mammalian cells.
And so the media are very simple that you need to.
It's just seawater.
Oh, that's it.
Yes, pretty much.
Oh, that's pretty simple, yeah.
Yeah. You know,
Okay,
we are using the capacity. So this macroalgae that we are using is quite particular, because it's a little bit of mixture, I will say, between plant and mammals in terms of metabolism. So it is able to perform the photosynthesis, and it is also having a urea cycle.
Okay.
But in our hand right now, we are using its capacity to grow with photosynthesis in order to reduce the.
And what is the nitrogen source? I mean, I guess that
It's ammonium.
Okay, yeah. That makes the media, I guess, even cheap, about as cheap as for E. coli, or maybe even cheaper.
Yes, yes.
Okay.
The media is nothing.
Yeah. Okay. And so for as far as the molecular biology goes, so how challenging is it to generate a stable clone, like a cell line for these, if you want to go for a new product, like compared to mammalian or even bacterial systems?
So it's in between both. It's a little bit longer than than doing some stable transformation in or transaction in bacteria, but it's shorter than doing it with mammalian cells.
Okay.
We are in between.
That is good. Okay. Yeah. So, any more questions from the audience? Fortunately, I don't see any hands up. Yeah.
Rita.
Yeah, Rita, very good. Yeah.
Yeah, yeah. So,
Take back.
well, I know already how microalgae is like, so like in terms of production of the culture media, as you said. But then you need to do some like extra purification steps because I also don't know how it is with the mammalian cells, so that's why I'm asking. So then it's
So we are accumulating the antibodies within the culture media. So we have a clarification step through filtration where we recover the culture media and remove all the cells. And then we do some affinity chromatography from the media. But probably we need to discuss with Nico and collaborate in order to,
Yeah, I mean, for
to be more
antibodies,
innovative in terms of DSP.
yeah, always happy to work on that.
I know.
Okay. So then I don't have any more questions. Yeah, I guess the audience is a bit quiet today. Yeah, but with that, then I guess, yeah, thank you both for coming and thank you for your great talks. And I also want to thank all the participants for joining. And yeah, then I will just invite you to next month's webinar. We will actually have Michael Sauer and Michael Eggermeier on the topic of biorefinery. Yeah, if you noticed, Michael Eggermeier's name was on Rita Mota's acknowledgement slide. So let's see what they will tell us. Maybe there will be some modeling in there too. Yeah, but then yeah, thank you all and have a nice afternoon. Bye.
thank you. Bye.
Thank you.